worked a couple of years there and quit my job, got back to IIT again for PhD, and developed a couple of technologies over there which can be used for diagnostics field, medical diagnostics. And now we are working dedicatedly to eradicate tuberculosis problem in India. Can you hear me? Just confirm.
Yes, sir.
Perfect. So today I'm going to discuss this one of the topic, which is development of a compact total internal reflection fluorescence microscopy module for the diagnosis of Mycobacterium tuberculosis. We have got Srishti 2018 GIITD Award with the help of BIIS for this particular project. So I have divided my presentation into two objectives. In first, I will take you through the fabrication and characterization of the device which we have made. It's a compact total internal reflection fluorescence device. So sometimes I will refer it as SeeTB, and the next objective of this presentation is going to be fluorescence sputum smear microscopy, which is used. It's a traditional method for tuberculosis diagnosis, and how we are actually going to use this method for TB diagnostics. So let's take what is microscopy and how I have I ended up developing this particular module. This picture is pretty old picture. You can see that our father of nation, Mahatma Gandhi, is observing leprosy germs in Sevagram ashram in 1940. He is using a very basic microscope. This microscope is being used for magnification of those germs and see the characteristics of physical characteristics of the germs he is going to observe. If you notice that microscope has two features. One, illumination. This illumination he is getting from the ambience light, and second is magnification. So every microscope has these two components embedded in that. These are like the basics. So either people have changed over the time the method of illumination, or they have modified the magnification. Nowadays, after capturing the image digitally, there are a lot of algorithms like artificial intelligence as well as image processing algorithms where you can refine the images as well. And we can achieve the sensitivity of single molecule. Molecule is the dimension of the molecule is in the range of angstroms. So let's go back to the history of microscope. You will see that the first microscope was milled in 1590, and you can see that the first microscope, it is a crude pipe where the lens is being fit in one end, and it was magnifying it. It was systematically got developed and getting better. In 1695, you will see that the more complex microscopy work started, and where they have put a systematic illumination of the object as well as observation. Why we need illumination of the object which we are observing? Because of mainly one thing, as the magnification will increase, you are collecting image or light from a very small area. So if you are taking light from a very small area, the chances of the photon you are collecting through your glasses or lenses are very less. So seldom or most of the time, the object looks very dark and you cannot see anything. So it is important to illuminate the object so that you can observe it better. In this endeavor, there are systematic evolution of the microscopy, and you can see that nowadays a simple microscope was developed, which you saw earlier in the picture I have shown. Then people reach to the next stage, which is fluorescence microscope, where the background has been kept dark and illuminating objects, fluorescing objects were observed. This microscopy helped breaking the boundary of resolution, which was not which was limited in simple microscope, was easily observable in fluorescence microscope. In order to refine it further and reach the higher resolution, axial resolution, and spatial resolution, more advanced microscopy methods like confocal microscope, total internal reflection fluorescence microscopes are being developed. You can see that, as the complexity of the microscopy is increasing, the setup is getting complex and bigger and bigger. So when I was studying this problem, and I was trying to develop a setup for our own laboratory in IIT Delhi, I was trying to understand that why this complex and why this bigger infrastructure is required if you have to observe the simple thing. Uh, then I come up with the basic concept of total internal reflection, which I learned in during my sixth standard or seventh standard. So this total internal reflection, everybody knows that when the light goes from high refractive index medium to lower refractive index medium, it goes away from the perpendicular. As you can see that n1 is the higher refractive index medium, n2 is the low refractive index medium. You can say that the glass coming light coming from the glass emerging out in the air, it goes away. If you keep on increasing the angle of incidence, you can see that one time it will come where the light will emerge as the parallel of the interface, and if you keep on further increasing the angle, you can see that the light gets totally internally reflected. So what happens the other side if light gets totally internally reflected? There are some electromagnetic disturbance which percolates a few nanometers of distance on the other side or lighter medium side, and it generates a electromagnetic wave over there. This wave is called evanescent waves, and total internal reflection fluorescence microscopy use these waves in order to enhance the resolution, axial resolution of fluorescence particles or fluorescence objects. So I tried, generating total internal reflection by taking a slab. So you can see that, I took the slab. So one side it was air, other side it was also air, and in the middle it was a glass. And I put the light. It went in and came out. So I kept on, increasing the angle, but light always, started coming out. And I was thinking that, how can I trap this light inside this glass slab? It was difficult. Then I checked that how these people are doing, who are making the microscope. So they said that you can achieve this microscopy by two methods. They use the prism, dove prism, where in between the glass slide and the dove prism in the first figure, left hand side, you can see that the specimen is sandwiched. The light is being incident from the side, and it gets totally reflected other side, and it illuminated the object over here, which was observed beneath by the 60x microscope, 60x objective. Or other method was the light which was illuminating the object came from the objective itself, which was shifted from the center to a little on the periphery, so that the light can emerge out and gets totally reflected within glass slab itself. And this glass slab and the material which is being used to couple, which is an immersion oil, has the same refractive index, so the specimen gets illuminated, and that is how they were using the fluorescence microscopy. So I thought if there are any other methods which can be used to reduce the setup or reduce the size of the setup. So I checked that there are a couple of literature published in order to make it more convenient for their experiments. So I saw that this TIRF lab technology, where they were using the thick glass slides and trying to put a light from one side of the slide, which gets trapped and goes into the zigzag path and generates evanescent waves on the top, where they are performing all the experiments, and in the bottom they were using the immersion oil. But the immersion oil and and the glass slab has a different has to be different refractive index. Otherwise, this ray will emerge out into the immersion oil. So they used a coating material, and this setup although generates a good total internal reflection. However, it's a difficult one to use it in general. So it is a very specific setup for a specific experiment. Similarly, other people have done it, where they have used thick glass slab, illuminated it from the side or in the circular slab, or they have chopped the slab and put the laser from one side. So I thought, when it is possible, why not design a device which can which goes universal to all the microscope if possible. Similarly, you can see that other work in which they have used objective to focus the light from one side, or they have used a very fine laser to illuminate from one side. So this was the literature which was available. So I started calculating that what is the critical angle for glass air, glass water, because this is what we are required in order to generate total internal reflection. And I learned that how this evanescent waves degrades as it goes inside, perpendicular to the axis. So these were the empirical relationships which I worked on, and found that if I put this incident ray close to the critical angle of either glass air or glass water, that is going to give us the maximum penetration depth. So I started working on the fabrication of the device. So I looked at the commercial TIR microscope, which was procured in the lab, approximately 1.5 crores, or approximately 1 crore. And I started making using a fluorescence microscope. I started making using a few lenses, my own microscope, and the device looked something like that. It was still bulky. I had to manipulate a lot of light, focus, defocus, and put it into a specific point in the objective lens, so that I can manipulate it. But it was not heavy side, because whoever was getting inside the room was had the hazardous situation of getting the flash in their eyes, or it was bulkier to use. So I thought, why not make it more compact. And then in the laboratory I made this setup where this laser has been manipulated in a particular way. There's a prism here which I've used and generated a waveguide. You can see that the clean waveguide has been generated, which is zigzag light which is coming in and out, and emerging out from here. And then I designed something where I put the prism, and cylindrical lens and a collimating optics, and put the light from the side. And I learned that it is getting trapped easily. It is generating high depth evanescent waves on the upper and lower side of the glass slide. And bingo, this was my setup. But another challenge that I have generated this kind of waves. I have shown it that these are getting generated. They are not getting percolated, even if I am putting the water, which is refractive index of 1.33, and refractive index of the glass is 1.5. So they are pretty good side. However, to put all this setup into a cabinet or device was a challenge. So I started designing a cabinet around this setup, so that all the time this prism is in contact with this glass slab, and not only just in the contact, but in a certain mechanical pressure, so that this light does not feel that the air is in between, and goes as way we want it to go. And this should not be complicated to use by any of the normal person, or non-optical person, where they can use it casually. So I started designing this device. I ended up making a very small handheld, lesser than the size of the calculator setup. And if this setup is being put on top of the microscope, you are ready to use that microscope as a TIR microscope. And this evolved further, and I started using a plate in here. This got a little bulkier, so that the coupling between the prism and the glass slide becomes very smooth, and there is no needness of smoothness of a certain degree. You can use the general glass slide and start generating the evanescent waves. So this is how we have generated results in our lab. This is the simple microscope we are using, and this device is illuminating. We are not using any illumination from the microscope. You can see that few observation which we could do easily by using the SeeTB setup is we could manipulate many magnification objectives, because in the traditional TIR microscopy, because the illumination is coming through the objective, so you cannot change the objective. But here, since it is independent of the objective, so I can observe the same sample in 10x magnification, 30x magnification, or 50x magnification, or even further 100x magnification. You can see that the same field, if I keep on changing, it gets illuminated nicely, and you can see the difference earlier, and now each bead is visible with high signal to noise ratio. I have compared it with the traditional CTIF and CTIF plus scattering. I'll tell you that what is the CTIF scattering, because this device gave us a lot of variety to manipulate with the microscope. So this is the epifluorescence microscope which is being used, and these are fluorescence beads which we use to observe. You can see that when we use the perpendicular illumination, which goes all the way from top to bottom, it generates a good picture if you see it in a standalone. But if you go to the TIR microscope, you can see that it's a high signal to noise ratio imaging, where you can see the difference between the beads. Each edges can be visible, so the resolution enhances. And I have also removed the filter, which is emission filter, because it was not needed, because the illumination was the perpendicular to the observation optics. So you can see that the signal to noise ratio enhanced significantly if I collected the scattering waves as well. So this was few observation I observed and quantified and published it into the paper in journal, and it was widely accepted. You can see that how if the size of the beads, which fluorescence beads which we were using earlier, if we keep on reducing the size of these beads, how these beads were getting better illuminated as the size were getting reduced. I'll tell you the reason of why it was getting better illuminated and why it was not better illuminated when the size was beyond a certain size. It's because of the depth of evanescent field. You can see here that the depth of the evanescent field is few hundred nanometers. So if 2 micron bead is being put on top, not all the whole portion of the bead is getting illuminated. However, if the size of the bead is getting reduced, the illumination size or area of the beads is getting enhanced. So you can see that here only a certain part of this particular bead. If you look on the top, this is the top view. If you observe from the top, you can see that this area is getting illuminated. If you go to the 1 micron bead, this area remains the same almost. And if you keep on reducing the size, the whole bead is getting illuminated. And I have calculated, and based on that and the geometry I have used, I could find the penetration depth of my setup, which was 150 nanometers. If you add the error, I have achieved 200 nanometers as well. This was the first ever setup I had made and calibrated. For later on, I played along with the optics and wave plates and multiple polarizers I have introduced in between, and this penetration depth increased further beyond 300 nanometers. You can see that the illumination within the glass slide was not a problem. I observed it in different distance, and the illumination intensity remained unchanged till if we reached to 20 to 25 nanometers, which is a large area was uniformly illuminated within the glass slide. This is one of the biological sample which we have observed. This is a HeLa cell. Uh, Fluorescently tagged. It was observed during fluorescence microscopy. You can see that everything is illuminated. However, in this image, only the things which are in contact with the slide and very close to the surface are selectively getting illuminated. So, this was the observation where we were trying to understand that how this cell adheres to the system and moves around, and this actin protein when it moves, or mobility, how it keeps on accumulating to one place and moves to other side of the cell. So these are some of the images which we have calculated and observed using the SeeTB device and compared it with the epifluorescence microscope. The interesting phenomena which we observed with this device is this device can be used without emission filter. That gave us edge over traditional TIRF microscope. So this system became a little better than TIRF microscope because in this image you can see no particles. Why? Because we have used no non-fluorescence non-labeled nanoparticles, like magnetic nanoparticles. They were not fluorescently tagged, so if you illuminate them, they are not going to give you any fluorescence. However, if these particles are being observed using SeeTB setup, which we have made, and if we remove the emission filter, you can see that the scattering light which is getting generated from each nanoparticle are very nicely observed by using this device in the simple microscope. Why it is a challenge, taking out the emission filter from the system, because if you see this figure, you can see that the light source, it generates, it throws all sorts of light. If you remove the emission filter, then the chances of getting the light which is illuminating this object will also come to the detector, and it will create a lot of interference. However, in our setup, you have seen in earlier slides that the illumination is perpendicular. I'll show you that again. This illumination is perpendicular, and observation is from top and bottom. So this light is coming from this direction and going out from this direction, has no interference in terms of the emission light. And so it's a clean signal which we can observe using the without excitation or emission filter as well. If we move on further, how this setup can be used further in science. So it can also be used for nano analysis of the gold nanoparticles, or maybe single molecule study. So FCS is one of the fluorescent correlation spectroscopy is one of the method which is being used for observation of diffusion or diffusivity of the very small nanoparticles. We tried using our device. We tried observing some of the diffusivity of the gold nanoparticles, which were in the size of 10 nanometers, and were suspended over the water. So this was the setup. You can see that the gold nanoparticle suspension were kept on top of the glass substrate. This is the illumination setup which has been kept over here, and gold nanoparticle were in the suspension. The light which has been put generated evanescent waves, and we observed from the bottom using long working distance objective, and we focused it very close to the surface interface of the gold nanoparticle suspension and the glass substrate. You can see that Some particles are getting eliminated, other particles are beyond this elimination zone, so they are not getting any interference. And very easily I can see that only few particles which are coming to the focal plane are generating the signal. This is the movie. If you look from the top, you can see that like the stars, whichever particle is coming to the focal plane are generating scattering signal, and whichever is far are only creating the background, and there's no change in the background. So you can see that various phenomena, all these gold nanoparticles are popping up and coming down. However, making the analysis of such big area is not possible because it requires a lot of computation power, and our computers, even the best of the computers, takes a lot of time to process this big image at once. So we took a small part of that, and we observed that since it's a very small part, so you only see the variation of intensity in the pixels, not precisely that how nanoparticles are coming and going out of this plane. And when we analyzed, it is something like, if you try to understand, this is the nanoparticle at t is equal to t1 position, or you can say at time is equal to 0. As the time increase, it may be come inside the, this is the observable focal area, where it comes inside and remains in there for some time, and it goes out. And in actual picture, not the very small area picture. If you look, and this is the yellow circle is the imaginary area. You can see that these particles are coming inside this area and getting out in some time. So these particles of such areas were observed, and this kind of scattering signals were generated. And when we did the autocorrelation analysis, we found that the diffusivity or diffusion rate of these particles uh, calculated through our system, and through the traditional method, which is DLS method, are very much comparable. And this kind of setup costs one few very little fraction of the actual instrumentation which we use for observing diffusivity, or maybe the imaging of total internal reflection fluorescence microscopy. I will come to the cost part of that device which I have made later, but I just wanted to tell that a little intervention in terms of making our own things can make a big difference in the cost and performance, the way we are observing it. So we thought of using this device for diagnosis purpose. Why it is thought it was thought like that? Because the interesting phenomena this device does, it converts a simple microscope, a simple bright field microscope, which are easily available over the laboratories or different places, to a compact total internal reflection fluorescence microscope. Not only fluorescence microscope, but compact total internal reflection fluorescence microscope. So we thought of putting one of our devices in such bright field microscope, and thought that if we can use it for diagnosis purpose, then it will be a great intervention. Then we don't require high-end microscopy to procure in order to get into the fluorescence-based diagnostic system. We can use this particular device only, and the existing infrastructure would be good enough. And since we used this device for TB observation, microvaccuum tuberculosis observation, so we named it SeeTB, or Chhe Drishti in Hindi. TB is Chhe Rog in Hindi. So when we looked at the scenario of tuberculosis in India, or not only in India but also worldwide. The scenario, TB scenario, is a pretty dangerous one. You can see that despite of all the possible good efforts by governments and WHO, there are multiple number of cases which are being missed, or missed every time they are getting diagnosed due to the sensitivity of the equipment. And one such person who has been missed for diagnosis infects around 10 to 15 healthy individuals over a 1 year of time, while getting into the contact with healthy individuals in the community or not. The misdiagnosis means a patient goes to the diagnostic centers, and where he or she gives the sample. Once the sample is being observed, uh, he has or she has been said that they are negative, and the sample, and since they are negative, uh, they go to the community easily. They say that I don't have TB, but actually they are carrier of the TB and they are spreading it over. So our priority was to design a product which can rapidly diagnose it. Not very, it should not be very costly. Even a healthcare worker with a minimal training observe it, and it should have a high sensitivity of observation, uh, which was in line with the target product profile published by World Health Organization, that you should, whenever you're designing a diagnostic device, you should adhere to at least these kind of uh, characteristic to the device. The TB diagnostics infrastructure in India, if you look at that, most of the people, because TB is also being considered a poor man's disease, so most of the people who get infected by TB goes to the designated microscopy centers for their diagnosis done, first point of contact. And if you observe, and we went through the statistics, there are 13,886. Now there are 14,000 some number designated microscopy centers in India. And as per the population of India, you'll see that one microscopy center caters to approximately 100,000 or 1 lakh people per center, which this number is 50,000 in the hilly and tribal area, because the population is lesser over there. So this is the method which is being generally used. A patient goes there in the peripheral community level centers. They give their sputum sample. A healthcare worker takes out the sputum, makes a glass slide or smear in a glass slide as per the WHO guideline, and that glass slide is being observed in a bright field microscope. Bright field microscope generate an image, and the bacilli or the infected germs or bacilli are present only in a very few number, and they are being tagged with a pink dye, and that pink is very light, and it merges easily with the background. And most of the time the observation and the sensitivity of that kind of test is lesser than 60%. That means out of 100, 40 people goes misdiagnosed. This is when healthcare worker pays full attention. If healthcare worker is a little distracted, and if the sample load at the centers are pretty high, then this observation time gets reduced, and the false negative cases keeps on increasing. Once they have been said positive, are being recommended for DOTS treatment, and if the symptoms still persist, they have been recommended to go to the intermediate or regional level, district level laboratories. You will find that such laboratories have not only smear microscopy but also gene expert or molecular based diagnostic methods available to them. They are more sensitive. They are more. They are faster. It takes approximately 3 to 4 hours to get the result. However, they are costly, and not everybody can afford that. Government of India is subsidizing this at this moment, but the cost of per test cost after subsidy is approximately 1,800 rupees, and which is not possible for a rural person or coming from a low economic strata person to afford that kind of test. Another test is a culture based test where the sputum sample is being inoculated into the culture, but such culture test takes approximately 21 to more than 1 month to get any positivity or negativity. By that time, a lot of infection can be spread easily. This culture test, however, is the most sensitive gold standard test. Is being called gold standard test because it gives if even if one bacilli is present and alive, it will grow and give the result. However, the time which the culture test takes is pretty high. If these tests are getting positive, the therapy has been recommended to the patient. If the symptoms still persist, they go to the different central reference labs. These are the bigger labs. They most of their work is to not only just to cure but also take the record of the surveillance, how the things are moving in terms of how the disease is getting spread in India as well, in India, different places and rural. So these labs not only just go for a TB test, but also go for resistance resistance of TB tests, and where extreme resistance or multi-drug resistance TB is also being tested, and similarly some similarly after diagnosis therapy is being recommended, and all the fast and advanced equipments are available in these labs. However, the chances of getting false positive, few false positive is also here. So our main aim was to do something at this level, where peripheral level, where the first time patient comes and give their sample to the diagnostic center. If we can change something over here, then definitely the number of patients who are getting cure will increase, and we can break the vicious cycle of TB spread. So what we did, we used our unique illumination technology, which we have patented as well. And also we have developed a thinning method, because breaking down sputum is also a challenge. Sputum is a thick, slimy substance which has to be broken down in order to all those bacilli which are embedded inside the sputum to come out. And make the whole system more homogeneous. Otherwise, what will happen that the one of the part of the sputum which we have taken might not have even one bacilli. Rest of the sputum might have a lot of bacilli, and still the test will come out negative, despite of person is being full of tuberculosis infection. So we have also developed a thinning reagent, which we named it CLR reagent. It's a proprietary reagent. Patent has been filed for that as well, which makes the thick, slimy sputum sample to homogeneous, transparent sample where the bacilli are equally distributed all over, so that it doesn't matter which part of the sample you take, the result remains unchanged. With the help of these two technologies and our compact design, uh, we thought that we will be able to make a nick in terms of getting the observation done. So you can see that this is I'm coming directly to the results. This is been the result which was observed using the fluorescence microscope, and we have used our SeeTB system, which consists of the thinning reagent as well as illumination device, and there is no change in the result. In fact, if you notice here, some of the bacilli are better visible here due to the high signal to noise ratio, which was earlier not very much. They are visible here, but the signal to noise ratio is not that good. I would like to take another bigger large area image of the bacilli. You can see that this here there are some background signal because there is a high LED or laser is illuminating the whole sample. In SeeTB, because it's a perpendicular illumination, there's absolutely no background. You can see that the dark everywhere, and bacilli are very easily visible. So the chances of getting bacilli detected over here is far better than the earlier traditional methods, especially the bright field method which is being used. So this is what I was saying, that this is bright field Ziel-Nelson test traditional method, which is being performed in the bright field microscope. And WHO said that it's better to use fluorescence microscope and use auramine O test because it has a better sensitivity over the bright field microscope. So people started moving from this to this, but every lab has this equipment. This cost approximately, this simple microscope cost approximately 20,000 rupees, 20 to 25,000 rupees. Moving from this simple bright field microscope to fluorescence microscope, if you take the cheapest one or the most economic fluorescence microscope, it costs approximately 2.5 lakh rupees. So from this to this is a huge infrastructure cost which has to be burdened by government and agencies which are planning to upgrade their existing microscope. However, what we did, we put our device, which is a very compact device. You can see that it is being put in the bright field microscope, and these are the results. You can see that the comparison. These are almost similarly observable here, and the cost of this device is approximately 20,000 rupees. In total, the total internal reflection microscope now costs you approximately 50,000 rupees. And within 50,000 rupees, you have a better total internal reflection fluorescence microscope available. This is the SeeTB process flow, which we developed over the time, where very small part of the sputum, 50 microliter of the sputum is being used, where CLR of the equal amount has been added, and let this sample to get homogenized for some time. This whole process takes 15 minutes. And after that, WHO recommended method has been used to make this smear, as well as staining and observation. We observed our sample using the SeeTB and simple bright field microscope, and this was the way of observing the sample, so that no bacilli has been, or no field of view has been left out, and all the bacilli would be visible. You can see that some of the results. Our limit of detection reaches to 10 to the power 4 bacilli per ml. Sometimes we have also observed 10 to the 3 cells limit detection as well, but for that I had to check more than 300 frames, and then we could see only one bacilli. So it's a pretty sensitive test, for a microscope. And if I compare my own SeeTB test with the existing fluorescence microscopy test, you can see that the bacilli which were visible over the SeeTB and epifluorescence are overlapping, and sometimes SeeTB could see few more bacilli which were avoided due to the high background by fluorescence microscope. You can see that how this the CLR reagent which we have developed works better, and how it makes enhancement in this existing SeeTB test. You can see this is the clearing reagent. This is a thick sputum smear. This is a thick sputum which is being collected, clean sputum, and when it has been mixed to different kind of bacilli load, you can see that how it behaves. This is the sample where most bacilli were present. It's been categorized as three plus TB sample, and this is 2 plus TB sample. This is 1 plus TB sample, and this is scanty. Scanty is like it's very hard to find even one bacilli if you check more than 500 field of view. So you can see that no matter how the sputum is, it gets very easily homogenized and almost transparent. So the reproducibility of the test keeps on increasing. This is the enhancement when we directly used it using the SeeTB. Or after that, we used it with the CLR liquefaction. In 3 plus sample, since there are enough number of bacilli present, so it doesn't matter if it is like liquefied or not. Each frame will have more than 500 or 1,000, maybe 1,000 bacilli present. Each area where we have to observe. But if you go to 2 plus or 1 plus, this number decreases significantly. However, with the help of CLR, since all the bacilli come into the homogeneous solution, you can see that the number of observed bacilli enhances. So there is a good number of impact of our CLR reagent which has been put, and we are aspiring that in further modification of this illumination as well as chemical reagent, we can reach more than 95% of the sensitivity in the test. This is the observation of TB sample where we have not put CLR reagent and where we have put the CLR reagent. You can see that although the bacilli are visible in these images, however, the background is also visible in that. This was used in fluorescence microscope, and this is in SeeTB. SeeTB, although the background has improved, however, some of the background is still there. But as soon as we have used the CLR reagent in fluorescence microscope as well as SeeTB, this background has been removed significantly, and only fluorescing object is bacilli. It can be very easily observed by any of the healthcare worker. It is easier when the background is black and the point which we have to detect is fluorescing. This is the clinical validation result which we have observed, where we have taken more than 237 or 250 samples actually, where few samples were not collected nicely, so they were not considered into the study. Only 237 samples were considered for this study, and we published that. You can see that all the positive samples which were observed by gene expert and liquid culture were also detected positive by our own system SeeTB as well, and the sensitivity of this SeeTB system came out approximately 90%, and the false negative false negative were also in the very less number. You can see there is a difference here in the liquid culture. number as well as in Gene Expert and in our sample as well, in our SeeTB as well, because there are non-tuberculosis microbacterium as well present in the sputum. These are although belong to the Mycobacterium family, these bacteria, but they are non-tuberculosis, so they does not generate TB. So since culture is the most sensitive and gold standard, it can pick all those numbers as well, which were not observed by Gene Expert, and these positives were also observed positive by our samples as well, because they get stain, and in the microscopy it does not make any difference whether it's a Mycobacterium tuberculosis or non-tuberculosis mycobacterium, because all the mycobacterium gets stained. This is the iteration where we generated or started building our device. We moved systematically from one to another based on the feedback which we were getting, or ease of usage, and make it more universal to all the microscope possible and easy to attach. Now, this device is the final device. This device does not need any attachment in the microscope. All you have to do is to just put that device on the top on the platform of observation part platform of microscope, and it is ready to use. And whenever you want to remove it, just hold it and take it out. That's easy it is to use nowadays. And the CLR reagent has came a long way as well. The reagent has been now made into tablet form, and although the stability of these tablets are under study, however, it is easier to transport these tablets, or transporting reagent in this form is much easier, and the usage is also very easy. If you take certain amount of sample, then you have to put one tablet or two tablet. It's been given in the description as well. So what we are trying to do next based on this technology, so we have achieved this. We have published our work in the Nature's in the Nature's journal, and what is the next thing which we are working on? So we started working on an automated artificial intelligence based fluorescence sputum smear microscopy device, which use the SeeTB system, CTF illumination, and CLR system. How we are doing it and why we are doing it is very important. Later on, when we were not working with the device and somebody else was working on the device, we found that there was a difference. There was a difference when an expert was observing the sample in the microscope, and a naive or not very much trained person was observing the sample, because of lack of experience or lack of experience, especially working with the fluorescence microscope and Mycobacterium tuberculosis. So what we did, if we can, we thought if we can remove the aspect where the healthcare worker should not be involved in terms of observing the sample, then it can be enhanced further. The sensitivity can enhance further, and the variation which generates by the human intervention can be reduced to minimum. So what we did, we designed an extensive, bigger device, which is highly automated, and it uses SeeTB illumination system and observation using a 40x microscope, all automated. This is a crude design of FACET TB. We named it FACET TB. FACET TB, you can see. However, the fancier one or the more product-like design has been generated now, and it will be available in some time. So what it does, it not only just observe or scan the whole slide which has been put. It also does the image processing, artificial intelligence based detection. But this does not happen all in the microscopy side. what happens is that there is an IoT module inserted here. So as soon as the microscope or the imaging device observe the image, it throws back to the central server, and where the image processing is being done. So this system becomes very light. It can be deployed in any of the rural place, and it can be communicated easily with the central server. All the data can be observed and processed over there, and the end result, which generates whether you have a certain number of bacilli has been present in each field of view or not, is being shown or sent back to the observer in terms of SMS or picture message. So this way, not only we are making the system more sensitive, but also adding a problematic surveillance aspect of that, because most of the time people does not know that how many patients are getting getting checked, or what are the number and outbreak in certain areas. So such kind of central server based surveillance system might also be helping them in terms of observing and better managing the disease. So you can see that how artificial intelligence is counting and making it more efficient. This is work in progress, and we are hoping to get this device more efficient device by next year, and so that it can be deployed to various centers. For our SeeTB device, we have applied for CDSO certification to put it for clinical validation and manufacturing, and also to selling. And we have got those certification, and very soon we are going to put this device not only in one center, which we are already using it, and we are going to use it for multiple centers for larger clinical validation study, and later on with the collaboration of the government and help of BIIS and ICMR, we are planning to fan it out for a larger geographical study, not across India. And we have also applied for ICMR project, and where ICMR committee has approved SeeTB to be used in MRHRUs, 4 different MRHRUs across India, and 1-year study will tell that how this system is impactful in terms of observing more number of sample than the existing setup, and with the minimum number of upgrading costs. So these are the conclusion to the SeeTB. The cost of deployment is pretty less, since the setup is can be manufactured in mass. So scalability is not a problem. Penetration, it can reach to the primary healthcare level. It's easy to use. Per test cost is lesser than 50 rupees. So in comparison to all the molecular tests, which are costing more than 800 or maybe 1700 rupees, can be this test can be used first. Only the person or patient who require the more sensitive tests can be recommended to those tests, and the design and the usability has been designed such a way that it can be easy to use to any of the healthcare workers. These are some of the research outcomes for our work, where we have published literature and filed patent. We have got the Guardian Young Technology Innovation Award for in 2018, and also Grand Challenge Exploration from Bill and Melinda Gates to work on this. And now we have got BIG for automated artificial intelligence based face-it device, and also IIT Delhi Innovation Award. These are some of the people which we are obliged to and grateful to in order to move in our journey, especially Dr. Anil Gupta, who has helped us, encouraged us. These are she is Deputy Secretary, Dr. Renu Sharoop, and all of the combined IIT community and BIIS always helped us in terms of moving forward. So that's it from my side. Thank you so much, and I am open for all the questions you have. I'm ready to take all the questions. Hello.
Good afternoon.
Yeah, good afternoon. Who is this?
I'm Ritvik, sir. In the second model that you're currently developing an AI-based identification system for tuberculosis, what
Yes.
would be the final cost and the scalability of that setup, sir?
That we are planning to keep it on the cost of existing fluorescence microscope. So cost has not been, we have not come to that stage where we are, working on the cost. First of all, we are working on to, make the system work and, fit well within the system. And later on, we'll come to the cost aspect, and we'll try to reduce that as much as possible.
Yes, sir. And in one of your tests that you have been conducting, you said there are a little false negatives that are observed using the technology. And what
Yes.
might be the reason of that false negative, sir?
There are a lot of things. So false negative could, I'll tell you that some of the false negative could be because of the artifacts. What happens that sometimes some of the background generates the signal which looks like bacilli, and it or maybe bacilli does not take the proper staining, so they are being not shown, or they are not being generating the signal, and it looks like that there are no bacilli in the frame. However, there are bacilli present. So some, because of error in terms of the healthcare worker is doing, or sometimes the way it has been prepared the slide, it's all human error, or maybe uh, some of the conditional errors where the stain is not reached or stain has not been prepared well. These are the reasons where we get few false negative as well.
Yes, sir. Another question is, the stain used is acid fast stain itself, sir, or is there any other stain that
Acid fast.
acid fast staining? Okay, sir. Thank you, sir.
Yes, it's WHO recommended auramine over staining only.
Okay, okay, sir. And so, did you test this, the addition to a microscope with the other microorganisms or virus particles?
Yes. Yes, we have tested it. However, we want to take one problem at a time. So we are taking tuberculosis as one problem, and that is what we are solving at this moment.
Yes, sir. And there is a virus particle which is of microns in size, and people generally say it is visible for light bright field microscope, but we cannot exactly identify that virus in light bright field microscope. Can that be a possibility of observation using this setup, sir?
Yes, definitely. There is a possibility of observing anything which is of bigger size or a smaller size, and if the stains are available, then it is definitely much better to observe in the fluorescence than bright field microscope. With the system, what we are selling is, or what we are giving is converting the existing simple bright field microscope into a high-end fluorescence microscope.
And like as you mentioned, we can use this for animal cell culture as well. You said HeLa cells were observed initially.
Yes. Yes, we have published our result, and HeLa cells we have used for to understand that what is the endpoint of adhesion and how the cells get adhered to the surface. So we used it as the way total internal reflection fluorescence microscope are being used to study all those cells.
Yes, sir. Thank you, sir.
Hello, sir.
Hello.
So my question is, in a normal fluorescent microscope, we use halogen lamps, mercury lamps for illuminating the fluorophores. Like here, we are using LEDs. So how far the sensitivity differs?
Right. It does not change anything. Nowadays, halogen lamps are getting replaced because halogen lamps have a life cycle. However, LED lights can be used for longer time without change into the watts and the power they are going to generate. So the sensitivity has nothing to do with the type of illumination. However, the source of illumination. However, the type of illumination makes the difference in the sensitivity. So you can observe here that in simple fluorescence or epifluorescence microscope, the illumination goes I'll show you the slide where it can be easily visible. This is the slide, slide number 28. Is it visible to you now?
It is visible, sir. Yes, sir.
Yeah. So you can see that this. Suppose there's a sample objective here. So this sample is crossing it through. This like in fluorescence microscope, in the light goes across the sample and illuminates every bit of that volume, cylindrical volume. However, in the case of total internal reflection fluorescence microscope, only the surface and some part of the surface is being getting illuminated, and that is where it is being observed. So you can see here that only certain part of the sample is getting illuminated, which is very close to the surface. That is how it is generating high axial resolution. In case of fluorescence microscope, it generates or illuminates everything here. So if sample is on top, it is also getting illuminated. And since focus can be done only one part, so the sample which is being illuminated on the top will generate only the background. So that is why total internal reflection generates high signal to noise ratio images. Did I make answer to your question?
Yes, sir. Thank you, sir. One more question. So what are the chemical ingredients in the clearing reagents?
Yeah, I will ask you to chemical ingredient to CLR reagent, right?
Yes, sir.
Hello? Yeah, so chemical ingredient of the CLR reagent would be mentioned in the publication which we have made. So if you go and in the Google type SeeTB, and you will get all the chemical ingredients in that. There's like a combination of, it's a cocktail of 7-8 chemicals which we have used in a certain percentage.
Yes, sir. Thank you, sir.
Any question now? If you want to ask anything, like if you want to discuss your idea, or you can also ask about GYTI to Dr. Vikas.
Yes, or maybe if you want to add on something on this technology later on as well, I would recommend highly to connect with me and work along with us in order to take it to the next level. Or maybe if you have some ideas based on this technology where you can use this, or maybe certain more technologies along with this technology. So I would be happy to help in that field as well. Whatever the idea is, and what Dr. Gupta has always pushed us to make the technology and idea available to all the people, all the young scientists, so that it can be enriched for the humanity.
So students, do you have any question? So I think I'll share your email ID, Dr. Vikas, if you don't mind, with the students. So later on, if they have,
that will be great.
Yeah,
they can connect.
Yeah. So Dr. Vikas, I'm really thankful.
Thank you. Thank you, Dr. Vikas.
Yeah, and this was really wonderful and very.
You're most welcome.
Yeah. Thank you so much.
I was happy to be part of that. Anytime. Thank you so much.